Thank you all for coming out. My name is Luke Bolstad, and I had the great opportunity of working under Dr. Kate Cooper in researching Dictyostelium, discoideum, multicellular response to various substrate stiffnesses. Now, in order to understand this research, it's important to know that we're all made up of cells, and they allow us to overcome extreme physical stressors, such as squatting a lot of weight or benching a lot of weight. But if you zoom into the microscopic realm of things, you may ask, what allows these cells to overcome physical stressors? So I invite you guys all to take out your notebooks and start jotting down some notes as we're about to learn some new vocab terms. The first of which is taxis. So taxis refers to the cell's movement response to various stimuli such as chemicals, light, and physical stressors. Now the cell's total encompassing process to how it responds to physical stressors is termed mechanotransduction. Now, physical stressors can manifest in many different forms, such as the cell being poked or pulled or experiencing shear stress generated by the substrate. And these are all present in the cell when it's migrating and when it's dividing. So mechanotransduction goes as follows. The cell senses physical stressor. Transmembrane proteins then transmit this signal to the nucleus, where it then impacts its gene expression, further having implications on the cell's behavior and the cell's function. So this is important for understanding how cells behave in disease states, and we will later learn that it's also important for understanding the cell's unique preference to geographical stressors, as there are many different types of geography in the body. There's many different locations, and they're all going to have their own unique physical stressors present. Now, I want to invite you guys to all time travel back in time with me to 1917, as understanding the mechanotransduction history is important for understanding why we care about it. So, in 1917, Darcy Wentworth Thompson published On Growth and Form, and this added to Darwin's evolutionary theory by not only stating that genes impact who we are, or who we are as an organism, but the physical environment also impacts the gene expression, which then leads to the identity of the organism. Mechanotransduction then came back into the spotlight in the 1950s with the introduction of the sliding filament theory. If you guys aren't familiar with this theory, it explains how you contract your muscles and the overlaying of the actin and myosin filaments within it. And then in the 1950s, we also had a cancer cell behavior discovery. So in this study, they took cancerous cells and non-cancerous cells, plated them onto a soft substrate or a stiff substrate, and they found that these cancerous cells were able to adhere to the soft substrate where the non-cancerous cells could not. And this gave us some motivation for understanding this research, as disease states are going to impact us as a human, and gives us a little bit of motivation for understanding this. So this led us to the discovery of the integrin protein, which is a transmembrane protein, and it is very important for sensing the extracellular signals and transmitting them intracellularly. And this led to our understanding of the role of matrix mechanics, or the extracellular environment, in tumor growth and formation in 1995. Current mechanotransduction research is centered around stem cells, or mesenchymal cells. These cells can differentiate into many different types of cells in the body, and as you can imagine, each part of the body is going to have its own geography and its own unique physical stressors present. So each of these many cells are going to have its own preference for what physical stressors it can handle and what physical stressors it may not be able to handle. And we see that in myocytes, which are muscle cells, and fibroblasts, which are fibrous tissue cells. And in these experiments, they plated these cells onto various substrate stiffnesses, indicated by the elastic modulus, or the gel stiffness, on the x-axis. And then they measured the cell's mechanotransduction response to the substrate stiffness, indicated by either striations or cell stiffness. And in myocytes, the percent striation is a hallmark of the cellular maturity. And we can see that they have a preference for what kind of substrate stiffness they want to be plated onto. They probably decrease their maturation or reaching their full potential after around 13 kilopascals, whereas the fibroblasts, they don't really seem to have a preference. They kind of exponentially grow and then plateau off. There's no decrease in the mechanotransduction response in fibroblasts. So different cells respond differently, have different mechanotransduction responses. So what's the big idea? I mean, this is in metazoan cells. Do other cells experience this? And it turns out that they do. And I will introduce you to the coolest cell on the planet, Dictyostelium discoideum. My pride and joy, our pride and joy. So this cell is a slime mold and it's found in the ground. So the next time you walk all over the ground, you can think, oh, there's Dicty down there. And they feed off of bacteria. And they have 3 different life cycles. So the asexual phase, or the vegetative cycle, these cells divide every couple hours, making them a very useful model organism to grow up in lab because they're cheap and you can grow a lot of them. The sexual life cycle consists of 2 opposite mating types fusing together to make an egg, which hatches future Dicty. And the most interesting part of the life cycle is the multicellular phase of Dictyostelium. So when these cells are starved and they don't have proper nutrients, they send out a chemotactic signal to other surrounding cells to get all the boys together to form this sort of aggregate that moves towards light. And the aggregate's called a slug. This slug then forms a fruiting body similar to a mushroom and then hatches spores, which then hatch into future generations of Dicty. And on this slide, we see a little GIF, or a GIF, as some may call it, of this process happening. Now, as you can imagine, these cells live in soil, and there's many different types of soil out there. There's sandy soil, rough soil. They're going to encounter different things in the soil, such as rocks and maybe some plastics, probably now. But they're going to have to change and elicit a mechanotransduction response in order to overcome these different soil types, as they're going to have different mechanical stressors present in each different soil. And a study kind of replicated this with single-cell Dictyostelium and their mechanotransduction response. So 3 different agar concentrations were made, or 3 different substrate stiffnesses were made, and they measured how stiff the cell was in relation to the substrate stiffness, and they actually found that on the softer substrate, the cell was less stiff, and on the stiffer substrate, the cell was more stiff. And this was just in the single cellular phase of Dicty. What about the multicellular response to this phenomenon? And the same study actually compared the 2 elastic moduluses, or the cell stiffness, of the slug versus the single cellular form, and they found that they respond quite differently. Now, this isn't only present in Dicty. This is also present in metazoan cells, or mammalian cells. And I want to draw your attention to this figure first, as we're going to be focusing on these adhesion proteins, or transmembrane proteins, that allow the cell to transmit signals from the substrate to the rest of the cell. And then these linkage proteins, which kind of act as, if you imagine 2 cells or 2 train cars, this would be the linkage between the 2 train cars, making up an aggregate, or a train, kind of. And in human mammary cells, aggregates can overcome a friction force generated by a substrate, but in the single cellular form, they could not. So this kind of hints at teamwork makes the dream work type of deal, that in the single cellular form, they couldn't travel, but in the multicellular form, they could travel. And in human fibroblasts and kidney cells, they dispersed when plated onto too stiff of a substrate. So this is kind of the opposite side of the spectrum. These cells are not able to handle the friction force generated by the substrate on too stiff of a substrate. And these linkage proteins are actually compromised, breaking the cells apart. And the multicellular aggregates can travel in larger groups on softer substrates. So this kind of adds to the previous point that I made, and this tells us that probably when plated onto too stiff of a substrate, these cells migrate in smaller groups, whereas on a softer substrate, those friction forces aren't going to compromise the linkage proteins. So this kind of leads us to our research question. What is the effect of substrate stiffness on multicellular Dictyostelium slug development? As the slug development of Dicty hasn't really been explored in the realm of mechanotransduction. And while investigating this, we could also ask what proteins are involved in mechanotransduction response to varying substrate stiffnesses. And to understand this, it's important to understand a single cell adhesion mechanism and how it moves. So the cell is going to place down a focal adhesion, which is in the purple. It's just a group of proteins that's going to kind of velcro the cell down to the substrate. And the cell is then going to use actin and myosin filaments to kind of rope itself along, pulling itself to the new focal adhesion. Then it's going to place a new focal adhesion, pull itself along. So kind of like a grappling technique. And this is true for single cells, but Dicty's a little different because they contain focal adhesions, but they're also shown to have van der Waals forces that are prevalent because they have a polysaccharide coat, which is a sugar coat on the outside of the cell. If you guys don't know what van der Waals forces are, they're small interactions between a polar molecule, so the negatively charged region is going to bind to the positively charged region. And they tested this hypothesis by plating these cells onto a plate and applying a current force to the plate. And they flooded the plate with different types of buffers, or different types of molecules, and they found that the nonpolar buffer wasn't as effective at detaching the cells from the plate as the polar buffer. So these polar molecules, or these charged, kind of charged molecules, are going to disrupt these van der Waals forces and detach the cells faster. So what about the multicellular phase? We have to pay attention yet again to these adhesion proteins and these linkage proteins, as these are going to be responsible for transmitting the signal throughout the whole entire cell. And this is just one layer of the cell. There's going to be many, many layers that are building on top of each other, and this signal from the substrate may dissipate over time to the rest of the cell. For the sake of this study, we're only going to be focusing on the focal adhesion proteins present in Dictyostelium, as it kind of makes everything a little bit simpler. And we see the evolution of adhesion machinery. And I want you to draw your attention to metazoa, which is mammalian, or us, on the left-hand side, or the right-hand side, and amoebozoa, which is Dictyostelium, on the right-hand side. And we see that integrin proteins, like we discussed earlier, are not going to be present in Dicty because they showed up on the evolutionary tree later after Dicty split from metazoa. But we do see some of these red proteins, such as paxillin, are conserved in Dictyostelium. So this leads us to the protein that we're going to be investigating, which is paxillin, as it is there's some domains that are conserved in both paxillin for humans, which is Pax A, and paxillin for Dicty, which is Pax B. And this study aligned both of the amino acid sequences of both human paxillin and Dictyostelium paxillin, and they found that they have conserved domains, which indicate they may have similar functions. And then they tested using a similar assay to what they did in the previous experiment that I discussed, where they plate the cells onto a plate and apply a current force to the Dicty to measure how fast these cells detach. And they found that those cells that did not have Dicty in them detach at a faster rate than those that did have paxillin in them, indicating that paxillin is important for adhesion and may be important for the mechanotransduction response of Dictyostelium. So to test this out, we took Dictyostelium cells, grew them up. This is a picture of them under a microscope. And we grew them up, plated them onto 3 different types of agar concentrations to mimic different substrate stiffnesses. And we then starved them to get them into the slug phase and put them in a light deprivation box so the slugs would move towards the light using phototaxis. After 20 hours were up, we then took pictures of the plates and measured the number of slugs formed on each plate, the distance of each slug traveled, and the length of the slug. And in the figure up here, we see what is classified by a slug and what is classified as not a slug. The Mexican hat is just it's when the slug settled down and starts to form that fruiting body or the mushroom type of the life cycle. And for down here, we have examples of the measurements of slug distance and slug length on the images. And we compiled all the data together, took the averages of each of the variables on each of the substrate concentrations, and we found no significance on any of the variables that we tested, but we did see some interesting trends, which I'll get into later. So for the wild type number of slugs formed in response to aggregate concentration, we didn't see any trends or any significance. But for the distance traveled by the slug, we did see a slight trend in increasing the distance traveled compared to the increasing of substrate stiffness. So this is interesting, keep this in mind. Again, for the length of slug, we didn't see any significance or any significant trends. And when moving into PAX B, yet again, see no significance or no trends available in the number of slugs formed in response to aggregate concentration. But we do see a kind of a similar trend to what we saw earlier in the wild type slug for the distance traveled by the PAX B slug. The length of slug, no significance and no clear trends. So when we compare the two, the wild type and the PAX B with their trends, we see that they kind of mimic each other. They have similar trends in that increasing the substrate stiffness will increase the distance traveled. Now it's important to note that we really can't make any clear assumptions about comparing PAX B to wild type directly because these experiments were not conducted on the same day. So that is something future experiments may have to take into consideration when they're performing their experiments. So some of the limitations of our experiment was there was temperature fluctuations in the lab. I don't know if you guys hang around the science hall too much, but it is awful with the temperature fluctuations, not very insulated. So we did have to throw out one trial in the wild type data that I would have liked to got back, but oh well. Another limitation is the agar concentration. We only tested a small range, so from 0.25% agar to 2% agar, and future directions could expand upon this by offering maybe an 8% agar. It might be impossible, but or a 6% agar or stiffer concentrations, just to give a larger picture of what is happening and what the thresholds are for this organism's mechanotransduction response. Another future direction could be investigating more protein mutants. We only investigated PAX B, but we were planning on investigating the following proteins right here, which are transmembrane proteins that have been previously proven to be important for the cellular adhesion of Dictyostelium. But unfortunately, when these plates came in, they were contaminated with bacteria, and we were not able to use them, unfortunately. So in conclusion, these trends may be useful for future experiments, but if we did everything right, then we know that Dictyostelium multicellular aggregates do not elicit a mechanotransduction response in terms of regulating their slug number formed, the distance traveled, or the length of the slug. And this is the same for PAX B. So future directions should take into consideration the trends, but no definitive claims can be made. With that, I would like to acknowledge all of these wonderful people on the board. Loras College Science Hall, you guys are like an extended family to me. Dr. Mosier, Dr. Edwards, and Dr. Kerr for jumping through some extra hoops to allow me to add this major and complete it in a year. And Dr. Cooper for taking me under her wing to perform this research. Nina Wien for being a friend to talk about Dicty with, super exciting. And my past, present classmates, thank you guys, and my family, thank you guys for all the support. And thank you for my dad for telling me to use my brain instead of my body when I was deciding on whether or not to go to college or not. Very happy with that. Thank you guys.